All right, well, I think we'll get rolling with the next presentation, which will be a bit of a change of pace, although I was happy to see Rob throw out some genetic stuff there, so I'm not the lone genetics nerd in the group. My work has been not so much focused on necessarily straight up ecology, and the major theme of today, but more of describing caribou units and how, once we resolve how we're going to save caribou, what are the best way to do that? Where do we draw our lines on the maps as far as which caribou do we decide we want to save, and what those caribou are? Like, where are they on the landscape? That is it the herd level, or is it something different? So, to start with that, we really, to start describing diversity in the landscape, we should have to start where before there was any diversity, so we go back to the last ice age. Glaciers covered most of Canada. Caribou were restricted to two places, in a Beringian refugium to the north and woodland habitat to the south. And this is where our two major subspecies of caribou in North America derive from: the barren ground tundra caribou in the north and our woodland caribou to the south. So glaciers melt, caribou populations expand, diversify, and according to morphological data and analysis, we get what we still recognize today as these number of subspecies in North America. Where woodland caribou are obviously dominant across the boreal region and mountain regions of Canada, and just dipping into the US, we have a Grant's caribou in Alaska, barren ground, and in some of the Arctic island populations. It's been covered, and so I'm not going to go into it in any detail, but we've also described the different ecotypes. And the new DUs have just come out, so this presentation is not going to reflect that, but it isn't necessarily going to change the results or how we view some of the recommendations. But essentially, until recently, woodland caribou described in three different ecotypes: boreal, northern mountain, and southern mountain. So what I've done for what I'll talk about today is consider 5 different types of caribou as far as from a genetic standpoint. I'm looking at the Alaska Grant's caribou, I'm looking at northern mountain caribou, southern mountain, boreal, and then barren ground. So some of the objectives of my PhD work in general have been included looking at this historical framework for evolutionary dynamics of caribou in Western North America, and sort of evaluate what do analyses reveal about the historic structure of caribou genealogies. So as we sort of evaluate what we've called different caribou, what do the genetics actually reflect and how caribou are different or the same? So where do they come from? We know generally either northern or southern source, but what have they done since then based upon changing ecology over time? And then how do these taxonomic designations relate to patterns of genetic variability? So how can we organize genetic differences into any meaningful categories such as subspecies or ecotypes? And are those categories meaningful for conservation? Some additional work I'm doing but won't have time to present include looking at some metapopulation dynamics and landscape genetics of caribou, specifically in Alberta, as well as equating all that sort of technical genetic work into comprehensible conservation ended information. So this is my sampling across Western North America. It's fairly robust. It's across like 5 different types, representing the 3 different putative subspecies and all the 3 different ecotypes. And briefly, we won't get into too much detail. I have a ton of samples, 854. I'm using 2 different kinds of genetics. Mitochondrial DNA, which really just broadly means that it's a slowly evolving marker, which makes it useful for looking at evolutionary scale questions. You're not going to be able to differentiate herds from one another using this marker, but you can theoretically differentiate at a higher taxonomic level, and that's what we use to reconstruct historic relationships among the caribou. We will also use the microsatellite data, which, in contrast to the mitochondrial DNA, are super useful for really looking at individual level relationships, as well as establishing, looking at rates of gene flow, and really sort of providing an individual level analysis of distinction on finer landscape scales. So getting right into the results and just sort of addressing really quickly some of the higher taxonomic questions is we ran a couple different models given sort of the a priori accepted designations of subspecies, and these models A, B, and C. What we're looking for is how well if we group using this specific kind of genetics, if we group our populations into what they're supposed to be, Grant's caribou in Alaska, or woodland caribou, or northern mountain, or barren ground. How well is the variation explained in those groupings? And depending how you group them, whether it's by subspecies, by ecotype, or by north versus south origin, we get some sort of level of explanation. If we start grouping them using some of the results from mitochondria, which is like southern mountain and boreal are quite similar versus what we found was northern mountain, which is considered woodland caribou, actually is more closely related to these northern groups of barren ground in Alaska. And so when we start looking at what the data is showing and reshuffling your organization, you find that current designations don't perform that well, and that these two a posteriori hypotheses explain diversity better. And I think an easier way to show this than the numbers is really plotting out population product comparisons, which essentially this is a plot of all the different herds they looked at and its relationship in genetic distance-wise to all the other herds analyzed. And you see a pretty clear distinction of, follow cursor here, of these are all of our southern mountain and boreal populations, which are indistinguishable as something Rob was mentioning earlier, the ecotypes not being distinguishable, and that's something we find at the mitochondrial level, versus in what we're calling a northern group, these derived from a Beringian origin, are grouping together separate from those, and then within themselves, these being Alaska caribou, these being northern mountain, and then our two barren ground populations. So one striking take home from this is that northern mountain caribou, which are defined as a mountain caribou, or as a woodland caribou, are actually more closely related on an evolutionary scale to barren ground and Grant's caribou. Now, whether this really matters in the end for conservation, that's to be debated, but it is, as far as we're giving out labels to these different groups and these populations, it could be important. Now, where we're getting more into a context on delineating management units, we're using the microsatellites. And we can see here again, these different K values really represent there's a statistical reason to use these. They're supported depending on the statistic you use. A K of 2 equals the microsatellites can significantly divide caribou in Western North America into two groups. And these two groups really represent our southern mountain boreal in one group, which is a southern origin prior to glaciation, or during glaciation, and then our northern group. If we look at K equals 3, we find organization into what we could say is putative subspecies, where we get southern mountains grouping together, boreal grouping together, and then northern mountain, barren ground, and Alaska again grouping into another group. And then where we start to get onto a more contemporary scale is at K equals 11. And we start to look at where we get populations like Little Smoky, which we were just talking about, that are assigned to themselves. And another group source, for example, Banff, Columbia North, and Jasper National Park are then assigned to within their own group as well, suggesting some sort of recent historic relationship among those herds that we now know through all of the monitoring and that telemetry data that these herds don't interact. Well, obviously Banff doesn't interact with anyone now, but that these relationships perhaps represent recent genetic exchange between these groups prior to the point of them getting extremely low numbers and to the point where they're not exchanging migrants between populations. Bringing this back to our recovery strategy that just came out for the boreal populations. Again, they were sort of classified as those in green being self-sustaining herds, those in blue important for connectivity, so effort needed to be made to save those, and the remaining populations in gray essentially being those that are not considered important, in other words, written off. If we overlap sort of our assignment tests, so these represent those bar graphs I showed previously on herds that are, or groups of herds that are have distinct genetic signatures versus what we're calling important herds versus those that can be written off. It doesn't really, on the right here, it doesn't really represent conserving, the amount of biodiversity within caribou that we're finding from the genetic standpoint. So I guess the take home here is that we are in a triage situation where we can't save everything, but we also are we going to spend a lot of money, time, and effort to save a herd for 50 years, and then without any context of what that herd is and what its relationship is to the rest of the populations? Or should we also, as we're figuring out what to save and how to save it, is important that it's not just West Athabasca, but this entire group historically had some sort of larger population dynamic that we have to, if you want to have caribou in perpetuity, greater than 50 years, greater than 100 years, we need to look at reestablishing these sort of connections and having caribou herds that are going to be self-sustaining. So I think I've covered all these summaries. I know we're in a rush, so I will just hand it over to Nick, and we'll wrap up after Nick with more questions.